Chapter 19, Chromosomes and Cell Division. There are two types of cell division in the human life cycle. There's meiosis and mitosis. Meiosis gives rise to gametes that have half the number of chromosomes as the original cell. This is termed haploid. In females, meiosis occurs in the ovaries and produces eggs, whereas in males, meiosis occurs in the testes and produces sperms. Mitosis, on the other hand, results in identical body cells that are diploid, meaning that they contain two of each of the number of chromosomes. And mitosis occurs during growth and repair of the body. Mitosis and meiosis, they differ. In the cells formed by mitosis are diploid cells, meaning that they have the two copies, whereas the cells formed by meiosis, the eggs and the sperms, are haploid, meaning that they have half the number of chromosomes as the original cell. This image shows meiosis between male and female. Meiosis is going to occur. The 2N represents two copies of each of the original chromosomes. So the male's gamete, the sperm, is produced in the testes, and this is going to take the sperm down to a haploid state, that N equals one set of chromosomes haploid. Meiosis is going to occur in the female, producing an egg. Again, the egg is going to contain one set of chromosomes, or haploid. So once the 23 from the sperm unite with the 23 from the egg during fertilization, this restores that diploid state, or that two sets of each of the chromosomes. This is termed into a zygote. A zygote is just a fertilized egg. After the sperm fertilizes the egg, everything after that is mitosis, which produces cells for growth and repair. Chromosomes are located in the nucleus. Their structure and function is that they are coiled, tightly coiled combination of a DNA molecule and special proteins called histones. These histones help with support and control of gene activity. Our DNA contains the genetic information which directs body development and maintenance. A gene is the term for a specific segment of the DNA. It directs synthesis of a protein which plays either a structural or a functional role in the cell. Somatic cells is the term given to all cells except eggs and sperms. Somatic cells have 46 chromosomes in humans, two sets of the 23 chromosomes. One came from the mother, one came from the father. Each somatic cell contains two chromosomes with genes for the same traits. These are called homologous pairs of chromosomes, or homologs. One chromosome of the pair is from the mother, and one chromosome pair is from the father. Diploid is the term given to a cell with two sets of chromosomes represented by 2N. Genes additionally occur in pairs on diploid cells. Members of each gene pair are located at the same position on homologous chromosomes, or on the same chromosome. Of the 23 pairs of chromosomes, sex chromosomes make up one pair. The sex chromosomes determine the gender, and there are two types, X and Y. If the individual has two X chromosomes, they are genetically female. XY designate genetically male. Sons will always inherit the Y chromosome from their father. Daughters inherit one X from the mother and the other X chromosome from their father. Autosomes are the terms that make up the other 22 pairs. These autosomes determine the expression or the phenotype of most of a person's inherited characteristics or traits. Mitosis is the process of cell division for body cells, for growth, and for repair. When one nucleus divides into two daughter cells nuclei with the same number and kinds of chromosomes, so mitosis produces identical cells. The cell cycle may take 16 to 24 hours to complete, yet only about 1 hour is actually spent in cell division. The cell cycle is a predictable set of events that a cell goes through from its origin to its own division into two daughter cells. There's two major phases: interphase and cell division. This image shows interphase, which runs from the growth phase G1 through the S phase DNA synthesis, through the G2 growth and final preparations for cell divisions. Mitosis itself consists of the phases of prophase, metaphase, anaphase, telophase, and then there's cytokinesis. So cell division runs from prophase through cytokinesis. Interphase is the longest of the two phases. Each of the phases of interphase, the G1, the S, and the G2, consist of cell growth and is characterized by the production of organelles, the synthesis of proteins and other macromolecules the replicated cell will need, and during S phase is when the DNA will be replicated. Interphase is the period of growth in preparation for cell division. It's not a resting period, it's very active, consisting of the three phase. The G1 is growth phase 1, S phase when DNA is synthesized, and G2 is the second growth phase. It does account for the majority of time that elapses during a cell cycle. During this time, DNA and organelles are duplicated, ensuring the daughter cells receive essentials for survival. Interphase, growth phase 1, first gap, it's a time of major growth before DNA synthesis begins, and the chromosomes consist of a strand of DNA called chromatin and proteins. Chromatin is just the disarrayed state of DNA. When DNA is tightly wound, as it is during the actual cell division stage, the gene activity is reduced, and so this is a control mechanism. But chromatin is just the disarrayed state of DNA, which allows during replication before it condenses during the cell division. Interphase, S phase, DNA is replicated, and the two copies of the chromosome are called chromatids, remain attached to the centromere. Growth is going to continue. G2 is the second growth phase, and it's the period after DNA is synthesized and before mitosis begins, and growth is going to continue. This shows the chromatin, the uncondensed form of DNA, the more disarray, whereas the condensed or duplicated chromosome is just this DNA in a very condensed state. So during interphase, chromatin is not condensed. It's in the disarray. You see it kind of looking like a unwound ball of string there in the bottom. During interphase, the two chromatids, they're not condensed. It's still considered chromatin. Mitosis begins DNA wraps around these histones, which the histones were the proteins that help regulate gene activity and provide support. DNA twists and loops around itself, and the duplicated chromosome consists of the two chromatids held together by the centromere. That's the little yellow between the blue chromosome representation. So the blue is representative of chromatid, the centromere is the structure holding these two chromatids together. Cell division consists of two processes, mitosis and cytokinesis. Mitosis is the term given to division of the nucleus. They'll go through several phases. Cytokinesis, on the other hand, is division of the cytoplasm and separation of the daughter cells. So DNA replication occurs during S phase, during interphase. Two sister chromatids are formed, banded together by the chromosome. During mitosis, these sister chromatids are going to separate and move to opposite ends of the cell, called their poles. And during cytokinesis, the parent cell is going to divide, forming two daughter cells, each containing two copies of each chromosome, or homologous pairs, or also known as homologs. The developing embryo and fetus, mitosis, still occurs. Body cells divide continually. Children, body cells divide in growth and repair. In adults, such as specialized nerve cells, lose their ability to divide. They enter a stage known as GO, wherein they carry out normal functions, yet they do not undergo mitosis and divide. Some cells, such as the liver cells, cease to divide but maintain the ability to undergo cell division in the adult should the need for tissue repair and replacement arise. In adults, our skin undergoes cell division continuously. Mitosis has four phases: prophase, metaphase, anaphase, and telophase. In prophase, chromatin condenses and forms chromosomes as the DNA wraps around the histones. The nuclear membrane will begin to break down and mitotic spindles will begin to form. In this condensed state, DNA cannot replicate and gene activity is turned off. In metaphase, these chromosomes attach to mitotic spindles and form in a line at the center of the cell. On the centromere, there is a region called the kinetic shore, and this is a region of non-coding DNA from which the mitotic spindles attach to the chromosome in order to pull the sister chromatids apart. But the centromere is the structure that holds the sister chromatids together. The kinetic shore is just a region of non-coding DNA on the centromere to which the mitotic spindle attaches. Alignment ensures that each daughter cells receive one chromatid from each of the 46 chromosomes. During anaphase of mitosis, the sister chromatids of each chromosome begin to separate, splitting at the centromere, holding them together. Now, separate entities, the central chromatids, are considered chromosomes. During telophase, the nuclear envelope is now going to form around each chromosome at each pole. The mitotic spindle will disassemble. This shows during interphase. During S phase, DNA is duplicated. Growth occurs throughout the G1, the S phase, and the G2, preparing the cell for cell division. In prophase, the chromosomes condense. Metaphase, they line up along the equatorial plate. Cytokinesis is the division of the cytoplasm and separation of the daughter cell. This begins in late telophase, and a ring of microfilaments contracts at the midline of the cell, eventually pinching the cell in two. So cytokinesis is division of the cytoplasm and separation of the daughter cells. This shows an image where a ring of the microfilaments contracts and constricts at the midline of the cell. These microfilaments continue to contract until the ring pinches the cell into two daughter cells. Each of the daughter